Alright, so how are you? I hope so that you all are doing very well, and we are starting the first chapter known as cell structure. So, the cell structure is the first chapter of your biology 9700 AS. Okay, so in the cell structure, there are basically you're required to acknowledge the two types of microscope, as you have already learned in your O levels. As the bracket was there, and in the bracket there was written that visible in light microscope or visible in electron microscope. So that was not the thing you're required to focus in your O levels, but yes, in your A levels there are the questions for your electron and light microscope, and you must know that what are these microscopes, resolution and magnification for. So the differences between plant and animal cells. This is what you learn from your primary classes, but now in your A levels and your AS level, there are something very difficult. Difficult means something very extended in the syllabus. So the plant cell and the animal cell being shown to you. All right. So in this plant cell, okay, the things visible to you are the tonoplast. Tonoplast is basically the membrane surrounding vacuoles. Okay, so first understand about what are the features in the plant cell you have already learned from the previous classes in your O levels or before. Okay. Cell surface membrane. You have already learned about cell membrane, that what is cell membrane in the plant cell. It is also present in your animal cell. Pressed against cell wall. It means that yes, there is cell wall and there is chloroplast in the plant cell, and there is a nucleus in the plant cell, and this is what you already acknowledged. Somehow you have already acknowledged about the grana. All right. So this is how you have learned in your O levels and in your previous primary classes about the cell membrane, cytoplasm, nucleus, cell wall, chloroplast, and grana in the plant cell. But now there is something very extended, and obviously this is not an easy level. So the extension of the syllabus means that now you're required to learn in more detail about everything, as now it is written as vacuole. Okay, so the sap vacuole is also you have already learned in your O levels and in your previous classes, which is the large with central position. Uh, it means that this is the large area in the plant cell, or the large surface called the sap vacuole, which is at the central position. Okay. There are mitochondria. What does mitochondria means? We'll be learning in this video. And in the nucleus now, what is new is that nucleolus, which is deeply staining, and there's the nuclear envelope. Nuclear envelope basically means nuclear membrane. All right. Why we called it nuclear envelope, we'll be learning in this video. And there's chromatin. Chromatin is very essential for you, even though the chapter number one cell structure is mean while it is very fundamental for you, and it is extremely required in your every of the chapter in your AS biology, it means that it is foremost essential for you guys. So do not skip this chapter and go through the YouTube and go through the other sections of the PDFs to just watch the cell structure and do more past papers, so that you can understand more about the cell structure, because in the cell structure most of the syllabus, most of the chapters from your AS Biology 9700 are related to the cell structure. Meanwhile, such as mitochondria, the nucleus. In the nucleus, there are chromatin, which is very helpful for you in your chapter number 5. Okay, if it does not acknowledge this in the chapter number 1 and more of the terminologies, the other chapters may confuse you. All right, so there are chromatin, deeply staining, and thread-like structures. Okay, and the tonoplast, which I was speaking at the first, it means that this is the membrane surrounding vacuole. So it is a simple membrane of the sap vacuole, means sap vacuole, and the membrane of sap vacuole called tonoplast. Okay, middle lamella is the thin layer holding cells together, contains calcium pectate. All right, this is the plasmodesma connects cytoplasm of neighboring cells. So this is the region called plasmodesma, which connects one cytoplasm to the other cytoplasm. And you can see that there is no cell membrane or cell wall. It means that they are connected. This region of the connection is called plasmodesma. Okay, cell wall of neighboring cell and the cell wall of the particular plant cell. Grana dust visible. We'll be also understanding the structure of chloroplast in this chapter for more understanding of the photosynthesis, which is another chapter. Small structures that are difficult to identify. These are ribosomes. Yes, these are ribosomes and the Golgi apparatus. So these are something very new and a bit confusing for you, but do not worry. There are a lot of explanation in this video. So I hope so that it maybe clarify you. Okay. In the animal cell, what is new for you guys? Wait a minute. Okay. So in the animal cell, just wait a minute to make it visible. Okay. So the cytoplasm again is what you have already learned in your O levels and in your previous classes. Cell surface membrane. This is what you have already learned. Okay. And the nucleus, you already know. So the nucleus and cell membrane and cytoplasm. Most of the questions already questions me that there is a sap vacuole in the animal cell. So if there is any sap vacuole in animal cells, so they are temporary, not permanent sap vacuole. All right. So that's why we does not even consider it. And this new thing is Golgi body. It means that Golgi apparatus. It's nothing new that above different from the Golgi body or Golgi apparatus. Both are same. Mitochondria are also present here. Small structures that are difficult to identify. These are uh, ribosomes, obviously. Okay, centriole, always found near nucleus, has a role in nuclear division. If you haven't watched the video of the live session of mitotic cell cycle, which is chapter number 5 of your biology AS 9700, so just go through and watch it where I was explaining about the centriole very in detail that what is the role of centriole in the nuclear division called mitosis. So this is essential as I was saying that this chapter is foremost for you guys, and if you skip this chapter or you just think that we'll be taking a lot of time to keep this chapter at the last to learning, so it may create a lot of problem because this is connected with all of most of your chapters. All right, so in the nucleus, the nuclear envelope or the nuclear membrane, the nucleolus and the chromatin are there. Here in the plant cell, what is new to you? Yes, the Golgi vesicle, the Golgi body, cytoplasm, chloroplast, middle lamella, plasmodesma, cell walls of neighboring cells, and vacuole, which consist of tonoplast as membrane or sap and a cell sap, mitochondrion. All right, and this is what important is smooth endoplasmic reticulum. What is endoplasmic reticulum? We will be learning in this video. Plus, there is cell surface membrane, ribosomes, which I was speaking continuously, nucleus, and rough endoplasmic reticulum. Microtubule. Microtubule is also essential for you in your nuclear cell division of mitosis. And there's a chloroplast which have the structure has been visible to you as grana and envelope. Envelope means basically the cell membrane. So this is the membrane of the chloroplast and a bit to it so that the figure may be clarified. All right, so this is what the structure is basically, and I hope so that this structure is very essential for you to learn. If you does not learn, because many times they give to just identify as A, B, C, or D, so it is very essential. As you can see that the mitochondrion, how are their structures like? All right, and you can see that in the smooth endoplasmic reticulum, this is the simple straight white lines, not straight lines but white lines, along white lines, while in the rough endoplasmic reticulum, you can see that the ribosomes are attached to it. All right. So this is the difference. The first difference what you have identified from the figure is that the smooth endoplasmic reticulum and the rough ER. All right. Okay, so that was one more thing is that this is what the plant cell and animal cell structures visible from the light microscope as written at the corner, and this is the plant cell visible from the electron microscope, and the animal cell which is visible from the electron microscope. One more thing is that you can see that in the plant and animal cell visible from the light microscope does not have a lot of structure. Meanwhile, the smooth endoplasmic reticulum and the rough endoplasmic reticulum plus Golgi vesicles, etc. But in the electronic microscope, a lot of things have been visible. It means that it is having a lot of great magnification and resolution. Now just focus on this animal cell, that what are visible in animal cell while using the electron microscope. Centrosome with two centrioles close to the nucleus and at right angles to each other. If you haven't started the chapter number 5 of your biology AS 9700, just go through the video which we have already done today, the live session. So the centrosomes function must be clarifying to you. What is microvilli? Okay, so the other name of microvilli is flagella, or we can say cilia. All right, as you can see, this is structure. So the microvilli means cilia, and the Golgi vesicles are visible to you. The Golgi body is visible to you. The microtubules radiating from centrosome. Microtubules I already taught you in the mitotic cell cycle. The ribosomes, cell surface membrane, cytoplasm, smooth endoplasmic reticulum, and rough endoplasmic reticulum, plus mitochondrion and nucleus. All right. Plus, what the new thing you're learning here or seeing here is lysosomes. So, what are lysosomes? You can see in this structure, I'm just zooming it so that it could be a great clarify to you. As now you can see that the lysosomes are larger than the ribosomes. All right. So now this is very clarifying to you. I hope that you will learn this and acknowledge the diagram, as this can be an MCQ to you guys as asking anything in this structure. And I hope so that nothing extra is required. All right, so now magnification. In your O levels, you have already learned about the magnification, that the formula for the magnification is image size upon actual size, or M is equals to I upon A. So this was the formula you was using in the first, in the paper number 6, or sometimes called paper number 4 of biology 5090 of your O levels. It was known as alternative to practical, or ATP, where they were been asking from you about the magnification of the figure, and you are required to use the formula of magnification, which was image size upon actual size, and the magnification was also written in the simple meters or centimeters. All right, but in your A levels, one thing is very essential for you to make in acknowledge is that the magnification is always measured in micrometer. It means that in A levels, I know that this chapter is the first chapter and first most difficult chapter for most of the candidates. That's why they just confuse themselves to the other chapters, not going through them and just starting the first chapter and keeping it to the 2 to 3 months. But it's not difficult at all. I know that magnification is a bit tricky, and if you go through the questions, they are a bit tricky, but you'll be scoring good marks when you'll be learning that how to have the unit conversions, which are very essential for you in your magnification. Although magnification is very easier for you, if you can see from the diagram and the scale of the eyepiece reticule, units have been given to you from the stage microscope, and the eyepiece reticule scale has been given to you. So then you can just measure it very simply, but the unit conversion may can lose your marks if you are not good in it. Okay, 1 millimeter is equals to 1,000 micrometer, and 1 micrometer is equals to 1,000 of nanometer. It means that first larger amount is millimeter, then the smaller is micrometer, and then the most smaller than micrometer is nanometer. It means that it means that thousands of nanometer is equals to 1 micrometer. All right. Resolution and magnification. The magnification means how much bigger a sample appears to be under a microscope that it is in real life. And the resolution means the ability to distinguish between two separate points. As resolution increases, image clarity and detail also increases. It is very easier and the simple words used in your daily life as resolution. All right. Okay, so this is a type of microscopes. So in the resolution of the light microscope is 200 nanometers. All right. And of electron microscope, so there are basically electron microscopes have two types of microscopes. One is SEM and the second is TEM. All right, so just wait a minute. I already know about that. What does this means basically? All right, so the TEM basically means that the transmission electron microscope, and the SEM means the scanning electron microscope. So the resolution for the scanning electron microscope is 3 nanometer and of transmission electron microscope is zero point 0.5 nanometers. And the magnifications are multiplied by 1500, and of electron is for SEM is multiplied by 250,000, and of TEM or transmission electron microscope is 500,000. All right. So it means that you're required to just multiply na, the magnification. Now moving towards the light microscope. So the light microscope is basically the limit of resolution is the half the wavelength. Ribosomes 25 nanometer. It means now just state it down in your notebooks that the what is the size of a ribosome is 25 nanometer, can't be seen with a light microscope as they don't interfere with the light waves. All right. So now you can see different stains are absorbed by different cell organelles, so they can be observed more clearly. B. Electron microscopes. Vacuum. Electrons cannot be focused without a vacuum as they will collide with air molecules and scatter. Water boils at room temperature in a vacuum, so that sample must be dehydrated. Specimen has to be dead. It means that the specimen for the light microscope is alive and for electron microscope is dead. This is the point. Just I request you to just write down the key points what I'm telling to you, because these are the MCQs. These are the basic MCQs appears in the paper. Advantage of a light microscope over an electron microscope is that can observe living tissues, more portable, easier to use, no technical training required, possible to see real natural colors and a live specimen. It means that the electron microscope have the black white images, and it can the light microscope can stain particular types of tissue for better visibility. All right, wait a minute. Yes. Okay. Cells are the basic units of living organisms. This is what you have already learned. The cell is a basic unit of all organisms. The interrelationships uh, between these cell structures shows how cells function to transfer energy, produce biological molecules, including proteins, and exchange substances with their surrounding. Now moving towards the electron micrographs of plant and animal cells. This is what you have already learned, is that nothing is new. Yes, this we have already learned from the above images that this is nothing new for you guys. Okay, so part B, eukaryotic cell structures and their functions. So number one. Okay, now what are the prokaryotes and the eukaryotes? Wait a minute. Where is the A written there? All right, just wait a minute. Alright, so basically, I will also read the book for you. Okay, as we are moving towards the cell surface membrane in the eukaryotic cell, so what are the eukaryotic cells? These are the animal cells and the plant cells. Alright, and what are the prokaryotic cells? They are known as bacteria. Alright, so what are the basic differences we'll be learning in this video? But before starting, we are required to just focus on the cell surface membrane, what is written in these notes and what is written in the book. I'll be reading both so that it could be more clarified to you. So, number one, uh, noted down that this is having the size of 7 nanometers, alright, and is made of phospholipid bilayer. Or you can say that the cell surface membrane is made up of um, phospholipid bilayer and the cell wall, which is present in the eukaryotes, are being made of fiber or the cellulose. All right. So, in the cell surface membrane, selectively permeable membrane that allows for the exchange for certain substances and barrier between cytoplasm and external environment and cell recognition or cell surface antigens, selection of substances that enter or leave cells. Alright, all cells, including those both of eukaryotes and prokaryotes, are surrounded by a very thin layer cell surface membrane. Alright, this is also sometimes referred to as the plasma membrane. Means the cell surface membrane is sometimes called plasma membrane. Okay, this is okay. As mentioned before, it is partially permeable and controls the exchange of materials between the cell and its environment. That's very simple. Okay, now just see the diagram of the nucleus, which is very essential. Okay, controls cell activities. Very dense, takes up color the most when stained and divides first during cell division, surrounded by two membranes known as the nuclear envelope, which is continuous with the rough endoplasmic reticulum. It means that it is continued with the endoplasmic reticulum, which have the ribosomes attached to it. Okay, as you can see in the diagram. All right. All eukaryotic cells contain a nucleus. The nucleus is a relatively large structure. It stains intensely, um, okay, and is therefore very easy to see when looking down the microscope. The deeply staining material in the nucleus is called chromatin. Chroma means color. Chromatin is a mass of coiled threads. The threads are seen to collect together to form chromosomes during nuclear division. Chapter number 5, what I taught you is mitotic cell cycle. Chromatin contains DNA, which stands for deoxyribonucleic acid, the molecule which contains the instructions or genes that control the activities of the cell. Inside the nucleus, an even more deeply staining area is visible, the nucleolus. This is made of loops of DNA from several chromosomes. The number of nucleoli is variable, 1 to 5 being common in mammals. One of the main functions of nucleoli is to make ribosomes. Now you understand what is the function of nucleolus is to make the ribosomes. Alright? Okay. It contains the nuclear pores allow and control substances entering in protein to make ribosomes, ATP or adenosine triphosphate, some hormones, nucleotides, and living mRNA or the messenger uh, ribonucleic acid. This will be learning in the video next. Ribosomes for protein synthesis of nucleus and the nucleolus, which is 2.5 micrometer, contains loops of DNA. Okay, several. This is what the ribosomes 25 nanometer. Okay, moving towards the ribosomes. Ribosomes composed of two basic units, two subunits, a small unit and large subunit to complete ribosome. Alright. Carry out protein synthesis. ATS ribosomes are present in cytoplasm and 70s ribosomes are present in chloroplast and mitochondria. Meanwhile, in eukaryotic cells, 80s ribosomes are present, and in prokaryotic cells, the 70s ribosomes are being present into it. Okay, and just wait a minute. Okay, so ribosomes are very small and are not visible with a light microscope. At very high magnifications using an electron microscope, they can be visible to consist of two subunits, a large and a small subunit. The sizes of structures this small are often quoted in S units, Svedberg units. S units are a measure of how rapidly substances sediment in a high speed centrifuge and ultracentrifuge. The faster they sediment, the higher the S number. Eukaryotic ribosomes are 80S ribosomes, and the ribosomes of prokaryotics are 70S ribosomes, so are slightly smaller. Mitochondria and chloroplasts contain 70S ribosomes, revealing their prokaryotic origins. Okay, and ribosomes are made of roughly equal amount by mass of ribosomal RNA, okay, and protein. Their three-dimensional structures has now been worked out. Ribosomes allow all the interacting molecules involved in protein synthesis, such as mRNA, tRNA, amino acids, regulatory proteins, to gather together in one place. All right. Rough endoplasmic reticulum. So, what is rough endoplasmic reticulum? Membranes that form an extended system of fluid-filled sacs, or it calls cisternae. Okay. Single membrane organelle ribosomes are attached to the rough endoplasmic reticulum and are the site of protein synthesis. Proteins made by the ribosomes enter the sacs and are often modified as they go through them. Small sacs, vesicles, break off from the endoplasmic reticulum and join to form the Golgi body. You can see this diagram for the more clarification what they are working for. All right, this is the maturing phase and the forming phase. All right, so that's all what is and the Golgi body. Okay, okay, all plus the rough endoplasmic reticulum. They can be found free in the cytoplasm as well and on the rough endoplasmic reticulums, the ribosomes. Moving towards the Golgi body or Golgi complex or Golgi apparatus, whatever you're required to say or whatever comfortable to you, just say it as Golgi body. These are the same words. Golgi apparatus or the Golgi body in a cell. This is shown to you, and I hope so that the figure is clarifying you. Yes. The Golgi apparatus is a stack of flattened sac called cisternae. More than one Golgi apparatus may be present in all cells. The stack is constantly being formed at one end from vesicles which bud off from the endoplasmic reticulum and are broken down again at the other end to form Golgi vesicles. The stack of sacs together with the associated vesicles is referred to as the Golgi apparatus or Golgi complex. Alright, what are the Golgi vesicles? Golgi vesicles carry their contents to other parts of the cell, often to the cell surface membrane for secretion. The Golgi apparatus chemically modifies the molecules it transports, such as sugar may be added to protein to make glycoprotein. Okay, so this is what the single membrane organelle packages substances into vesicle for transport glycosylation. All right, this is what about the Golgi body. Moving towards the smooth endoplasmic reticulum or SER. Synthesizes lipids and steroids such as cholesterol and the reproductive hormones such as estrogen and testosterone. The All right. So we were on the smooth endoplasmic reticulum. It synthesizes lipid. Okay. The smooth endoplasmic reticulum has a smooth appearance because it lacks ribosomes. It has a completely different function to rough endoplasmic reticulum. It makes lipid and steroids such as cholesterol and the reproductive hormones such as estrogen and testosterone. Smooth endoplasmic reticulum is also a major storage site for calcium ions. This explains why it is abundant in muscle cells where calcium ions are involved in muscle contraction. In the liver, SCR is involved in the drug metabolism. Alright, so this is not the great explanation written here, but in the book. Okay, so now what is basically important is lysosomes, which is 0.1 to 1 micrometer small in the size, is, wait a minute, alright, spherical sacs surrounded by a single membrane, not permanent structures, no internal structure, contain hydrolytic enzymes responsible for digestion, breakdown of unwanted structures such as old organelles, can even digest whole cells, such as in mammary glands after the period of lactation. Now you can see that this is the structure where the lipid layer being visible to you, the membrane and the hydrolytic enzyme mixture plus the glycosylated membrane transport proteins. All right. Energy releases from energy rich molecules such as sugars and fats during respiration is transferred to molecules for of ATP or adenosine triphosphate. ATP is the energy carrying molecule in all living cells. Once made, ATP leaves the mitochondrion and can spread rapidly to all parts of the cell where energy is needed. Its energy is released by breaking ATP down to ADP, means adenosine diphosphate, in a hydrolysis reaction. All right. Okay. Now in the book there is a very detailed thing written, which is not in the PDF but very essential for you guys. Okay, so the lysosomes are simple sacs surrounded by a single membrane. In an animal cell, usually zero point 0.1 to zero point 0.5 micrometer in diameter, and in a plant cell, the large central vacuole may act as lysosomes, although lysosomes similar to those in animal cells are also visible or also seen in the cytoplasm. All right, lysosomes contain digestive enzymes. Okay, so this is what we have already learned. Okay, getting rid of unwanted cell components. So the lysosomes can engulf and destroy unwanted cells components such as molecules or organelles that are located inside the cell. Cyto, the lysosomes have three functions. The three basic functions of lysosomes are endocytosis, exocytosis, and autolysis or the cell digestion. So, in the endocytosis is described in more detail in chapter number 4. Okay, we will also learning in that chapter, but in this chapter, material may be taken into the cell by endocytosis. Okay, for example, when white blood cells engulf bacteria. This is the example of the endocytosis. In the endocytosis, material may be taken into the cell by endocytosis, for example, when white blood engulfs bacteria. Lysosomes may fuse with the endocytic vacuole formed and release their enzymes to digest the contents. Exocytosis means the lysosomal enzymes may be released from the cell for extracellular digestion. An example is the replacement of cartilage by bone during development. The head of sperm contains a special lysosome called the acrosome for digesting a path through the layers of cells surrounding the egg just before fertilization. This is the example for the exocytosis. Also remember the examples, then you can understand the functions, or then you can understand the terminologies. All right, what is the autolysis? It's self-digestion. The contents of lysosomes are sometimes released into the cytoplasm. This results in the whole cell being digested, a process called autolysis. This may be part of normal development, as when a tadpole tail is reabsorbed during metamorphosis, or when a uterus is restored to its normal size after pregnancy. It also occurs after the death of an individual's as membrane lose their partial permeability. Okay, one of the example you can hear is that when the woman is pregnant, so then the placenta obviously is increasing in the size. So the size of the uterus should be towards back position in the original position or in original size. So this occurs autolysis, all right, or the self digestion after the birth of the baby, obviously. Okay, and what is a microtubule? We have already discussed microtubule in the chapter number 4 in the in the chapter number 5 of the live stream. If you have not watched it yet, I just request you to go and watch so that it could be very helpful for you. We are already detailed, have a detailed explanation about the microtubules. Okay, it is very essential for you for your mitosis. So the tubulin dimer is alpha tubulin and beta tubulin. Okay, and this is the cross section. This is microtubule, long rigid hollow tubes found in the cytoplasm made of a protein called tubulin. Tubulin has two forms, alpha and beta tubulin. Alpha and beta tubulin molecules combine to form dimers. Many dimers are joined end to end to form protofilaments. 13 protofilaments are in a ring to form a cylinder with a hollow center. This cylinder is microtubule. First, the protein alpha and beta join together to form dimers, and then dimers joins to form 1 uh, pro- protofilaments, and that for 13 protofilaments, which makes one microtubule, and it supports and gives shape to the cell. The assembly of microtubules from tubulin molecules is controlled by special locations in the cell called microtubule organizing centers, or MTOCs. All right. Okay, so what is centrioles and centrosome? This is what we have already learned in the video of mitotic cell cycle, as there are 9 triplets of microtubule making one centrosome. Oh, sorry, one centriole. Outside the nucleus of an animal cell, two centrioles are present close together at right angles in the region called the centrosome. Centrioles are hollow cylinders about 500 nanometer long, produces spindle fibers, organizes microtubule. Okay, now here's something very essential about his chloroplast. Okay, so just wait a minute. Okay, so we are at the chloroplast. So the visible things in the chloroplast, I hope so that this might be visible to you, is stroma, thylakoid, granum, outer membrane, inner membrane, and lamella. This is focus on this diagram first. As you already know about the grana and the lamella, about the stroma. The new thing is maybe thylakoid and lamella, and maybe stroma, and most of you maybe does not acknowledge the granum. All right. So the diameter of a chloroplast is 3 to 10 micrometer. Carries out photosynthesis. This is what we have learned in the O levels and in the previous classes. All right. Okay. Contains starch grains, cellulose DNA, and 70 S ribosomes, as we have discussed. ATP is also produced here. All right. Just wait a minute. Okay. What is the okay? So in the chloroplast, what is the function of the cell wall? Is gives cell definite shape and rigid as made of cellulose. Okay. So cell wall in the eukaryotes are made of cellulose, but in prokaryotes are made of uh, we have been learning in this video. Okay, so freely permeable. Okay, so the cell membrane is partially permeable while the cell wall is freely permeable, prevents cell from bursting. And plasmodesmata, this was the region which has continued one cytoplasm to another. Plant cells are linked to neighboring cells by means of fine strands of cytoplasm called plasmodesmata or plasmodesma, which pass through pore-like structures in their walls, allow the transport of water, sucrose, amino acids, ions, etc. between cells without crossing membranes. This is called movement through the symplastic pathway, allows communication signaling between cells. Okay, what are the vacuoles of? Vacuoles are surrounded by a partially permeable tonoplast which controls exchange between the vacuole and cytoplasm. As you can see this structure carefully, as there is tonoplast, central vacuole, and the cell. All right. Okay. Um, helps regulate osmotic properties of cell. Fluid present in the vacuole consists of pigments, enzymes, starch, organic molecules, mineral salts, oxygen, and carbon dioxide. As you can learn this as PASOMOC. This is the trick for you guys. Structure features of prokaryotic cells, organisms that lack nuclei or proper nuclear membrane are called prokaryotes. Unicellular 1 to 5 micrometer cell wall made of murine peptidoglycan. So this was I want to try to say that the cell wall is made of peptidoglycan and peptidoglycan, and of the cell wall in the eukaryotes are made of cellulose. So this is the difference. Okay, no membranes around organelles. 70s smaller ribosomes, genetic material in the form of circular DNA, have no ER. Okay, additional structures sometimes present and structures always present. So the always present structures are cell wall, cell membrane, cytoplasm, circular DNA, and ribosomes. Means now circular DNA. It is not in the nucleus, but it is free DNA. Okay. And what are the additional structures? Flagellum for locomotion, very simple structure. Okay, so what is cilia or what we learn in the microvilli is for? So this is for two things. One is for locomotion or the movement, and second is the larger surface area for absorption. Capsule, additional protection. Enfolding of cell surface membrane may form a photosynthetic membrane or carry out nitrogen fixation. Plasmid, a small circle of DNA, several may be present. Okay, and Pili is for attachment to other cells or surfaces involved in sexual reproduction. And at the last, we are moving towards what are the differences between the what are the difference between the prokaryotic and eukaryotic cells. So the size of the cell is in the prokaryotic cell, the size of the cell is 0.2 to 2 micrometer in diameter, while in eukaryotic it's 10 to 100 micrometer. Means eukaryotes are larger. Examples are in eukaryotes, the examples are animal and plant, and in prokaryotes are bacteria and archaea. Okay, the nucleus is absent in prokaryotic cell and present in eukaryotic cells. All right, flagella. Prokaryotic cells consist of two protein building blocks, and in the flagella, which is complex, consists of multiple microtubules in eukaryotic cells. Cell wall usually present chemically complex, all right, in prokaryotic cells, and in eukaryotic cells only in plant cells and fungi, chemically simpler. Obviously it's not present in human body. The cell wall. Okay, the plasma membrane with steroid, usually not present in prokaryotes, but present in usually not present in prokaryotes, but are present in eukaryotic cells. The cytoplasm, no cytoskeleton or cytoplasmic streaming is present in the prokaryotic cells, while in eukaryotic cells, cytoskeleton or cytoplasmic streaming are being present. Ribosomes are smaller in prokaryotes, are of 70s ribosomes, and 80s ribosomes are larger in eukaryotic cells. The cell division in prokaryotic cells are binary fusion, and in eukaryotes are mitosis. Number of chromosomes more than one in eukaryotes, and ones but not true chromosome in prokaryote. And at the last, sexual reproduction so involves meiosis in eukaryotic cells, and no meiosis, transfer of DNA fragments, only conjugation, okay, in the prokaryotic cells. This is what the we are required to learn, is that prokaryotic cells as well as eukaryotic cells are covered with the plasma membrane, which is located on the top of the cell membrane or mucus capsule. Despite of its relative simplicity, prokaryotics are typically independent cells. Table 4.1 present the major difference between the prokaryote and the eukaryotic cells. And at the last, we are moving towards the diagram of the virus. So as you can see that the virus having only three things. One is DNA or RNA, genetic code. The second is plasmid, and the third is protein molecules. Nothing extra is here in the virus. It is non-cellular, or you can use the word acellular. Okay, protein code. Okay, nucleic acid core, DNA or RNA strand, and replicate inside host cells only. Means it is not considered as a live cell, but the science is still considered it as a life because of its reproduction, that it can only reproduce while it cannot breathe or can have any function like the living organisms above than reproduction. Show no characteristics of living organisms, symmetrical shape. The virus DNA RNA takes over the protein and synthesizes machinery of the host cell, which help to make new virus particles.